That's right, you just turn the camera off now, right? So everybody's here. Oh, come on, monitor. This is ridiculous. Source, computer. There we go. All right. So, yes, glycolysis, the final frontier, right? So, one of the things I want to just say before I get started is that with glycolysis, we see a lot of molecules, we see a lot of enzymes, a lot of reactions. But I think it's important, and one of the way I usually teach this is I will go through the reactions and so forth with you, and I'll give you a little perspective on each of these. But after we've seen the pathway, okay, then you can start to understand some things physiologically, how these things make sense, okay? So going through the pathway is a necessary evil. We have to get through the pathway to see the overall big picture. But after we've gotten through that pathway, and in fact, as we get to a couple of the reactions in the pathway, you're going to understand something different about glycolysis or about the burning of sugar than you knew before, okay? And it's not just the fact there's a bunch of names that I gave you, okay? So let's go through that and pick up where we left off last time. Last time when I finished, I had basically gotten to the formation of the fructose 6-phosphate. And fructose 6-phosphate is, fructose 1,6-bisphosphate is a fairly high energy molecule. Okay? And it's a good thing it is, because the next reaction that happens is a difficult reaction. It's a very difficult reaction. And one of the reasons it's difficult is you basically have to chop this thing into two pieces, meaning you have to break two bonds in this thing. So you've got to break two bonds, and in breaking two bonds, you end up with two molecules. Okay? This reaction in glycolysis turns out to be very unfavorable. Very unfavorable, meaning that if I start with equal concentrations of products and reactants, the delta G will be positive, meaning that the delta G0 prime is positive. Okay? So how does this guy go forwards? Right? How does a reaction go forwards when it starts out with a delta G0 prime that's very positive? Okay? Well, I will take you back to the equation that we've talked about, which is delta G equals the delta G0 prime plus RT times the log of products over reactants. So we know that the concentration of products and reactants can change, right? What happens if I start removing products? What happens to that ratio of products over reactants? So the ratio gets smaller, right? And the logarithm term becomes more negative, right? What happens if I start dumping in a bunch of reactants? Same thing, right? So now I see that ratio gets even smaller, and so the smaller that ratio gets, the more negative the log term gets. And if the log term gets negative enough, what happens to the delta G? It becomes negative. Now, what I've just described to you is a very important principle that we see in metabolism known as pushing and pulling. Okay? How do I push a reaction? Well, if I want to push a reaction, I put a lot of reactant in. So by increasing the amount of fructose 1,6-bisphosphate, I push the reaction forwards. How do I pull the reaction? I pull it by removing the products. So the combination of pushing and pulling helps this reaction to get over an energy hump. And as we will see, the cell pulls a very, very cool trick later on that allows this reaction to go forwards. This reaction is really important, okay? Because if this reaction doesn't go forward, then all glycolysis is hosed. And this is the most energetically unfavorable reaction that we have in glycolysis. Okay? All right. So the enzyme that catalyzes this is the only enzyme whose name doesn't tell you directly what it does. It's called aldolase. A-L-D-O-L-A-S-E. Okay? And the products of this, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, are important cellular intermediates in several pathways. Okay. Well, the very next reaction we can see what happens. This reaction, the dihydroxyacetone phosphate is actually converted into the glyceraldehyde 3-phosphate. And that turns out to be convenient for memorization purposes because from this point forwards, we have 2 of every molecule that goes forwards. So now we have, starting with glucose, we've gone all the way down, and now we will have 2 molecules of glyceraldehyde 3-phosphate. The next step, we're going to have 2 products, et cetera, going through the pathway. This enzyme turns out to be a really interesting enzyme. It's called Triose phosphate isomerase. And this enzyme is an example of an enzyme that is what I describe as perfect. What's a perfect enzyme? It goes as fast as it possibly can, okay? And the only limitation for a perfect enzyme is how fast the substrate can diffuse into the active site. This is the first enzyme that we've talked about in class that is perfect. We cannot make it any faster because we can't make diffusion happen any faster. Well, why is this enzyme perfect? Why is it perfect? It's perfect because this enzyme has an intermediate in the process that is very unstable. So to keep the intermediate from falling apart, the enzyme has evolved the ability to make this reaction go as fast as it possibly can so that very little of the intermediate gets a chance to fall apart. So in this case, making it go really, really fast improves the efficiency of the reaction immensely. Okay. Well, so now, and the good thing for us, as I said, we have 2 molecules of glyceraldehyde 3-phosphate. Yes, Lisa?
You said it's going fast, so it stops the dihydroxyacetone reacting?
No, no. So there's an intermediate that's not shown on here, and going between. By the way, you can call this DHAP, and you can call this G3P. Okay. So there's an intermediate in between the two. En route to that, that's the unstable thing. So rather than have a slow process where that intermediate sits there for a while, the enzyme has evolved a process where it makes this thing go very, very fast. Okay, so DHAP and G3P. 3P, that's right. Okay. So at this point, we've finished the first phase. Now, to summarize in the first phase, what did we do? We took glucose, we put a couple phosphates on it using an ATP each time. So we've already burned 2 ATPs. We haven't gotten any energy out yet. But now we're getting ready in the next phase. We're going to get some energy. We're going to get some ATPs back. And we're also going to get some NADHs, which will happen in the very next reaction. Okay? Well, at this point, we remember now that we have 2 molecules of glyceraldehyde 3-phosphate moving forwards. And this reaction is a very interesting reaction as well. All right? Look at what's happening here. We start out with a molecule that has 1 phosphate, and we're making a molecule that has 2 phosphates. Now, when we did this with glucose and we put a phosphate on, what did I tell you the cell had to do to put that phosphate on? It had to use an ATP, right? And the same thing happened when I tried to put a phosphate onto F6P to make F16BP. I had to use an ATP. But here I've put a phosphate on and I haven't used an ATP. Why am I able to do that? What's different about this reaction than occurred in those other reactions? Yes?
It has the ADH subconjugate.
Okay, so when we see NAD and we produce NADH, what does that tell us? There's an oxidation that's occurring. Oxidation provides some energy. So we don't need to make or use an ATP. We use the energy from the oxidation of the glyceraldehyde 3-phosphate to make this happen. So there's actually 2 things that's happening here. All right? Notice we started with an aldehyde. This is an aldehyde. Over here, this is an ester. The only way we have an ester is if we have an acid. So we've actually oxidized this thing from an aldehyde to an acid. All right? So there's an oxidation that's occurring. and there's a phosphate getting attached. That's the two things that's happening in this reaction. This is the only oxidation that occurs in glycolysis. It's the only oxidation. Okay? We're making a molecule called 1,3-bisphosphoglycerate, which you're probably going to call 1,3-BPG. And that sounds like something you've heard of before, but it's not the same thing. That'll come up in a minute. Okay? All right, so the enzyme that catalyzes this is known as glyceraldehyde 3-phosphate dehydrogenase, or if you want to call it G3PDH, that would be okay. G3PDH. Now, as I said before, frequently when we have an oxidation, we will have a dehydrogenase involved, and that's exactly what this is. It's a dehydrogenase that's catalyzing an oxidation. Okay. Now, this step is the first time that cell is going to make an ATP. The first time that the cell is going to make an ATP. Remember, we're starting again with 2 molecules of this because we have 2 of everything going forward. So we're going to make 2 ATPs. At this point, we will have broken even on the ATP business. What's happening here? Well, these 2 phosphates are very, very close to each other. They're very negatively charged. They very much don't want to be next to each other. And in fact, they repel each other. So it becomes easy to take one of these phosphates and put it onto ADP to make ATP. In fact, I showed a table on Wednesday that showed you that this molecule, 1,3-bisphosphoglycerate, actually has more energy than ATP. I asked you at the time, I said, how is it that we can make a molecule that has more energy than ATP? You've just seen the answer for part of it. Oxidation made that possible. And now the energy of that molecule is being used to transfer a phosphate onto ADP to make ATP. This is an example of a class of reactions that you should know the name of. Okay? The class of reactions are known as substrate-level phosphorylation. Substrate-level phosphorylation. Now, substrate-level phosphorylation occurs when a high-energy molecule transfers a phosphate to ADP and makes ATP, just like this. The reason I want you to know the name of this is that there are only 3 ways in cells in which we make ATP. One of them is substrate-level phosphorylation. The second one occurs in your mitochondria, and we'll talk about it next week. It's called oxidative phosphorylation. Actually uses oxidation to make ATP. Okay? We used oxidation to make 1,3-bisphosphoglycerate. We didn't make ATP with oxidation here. Okay? All right. And the third way in which we make ATP, we don't do, plants do. Photophosphorylation. They use the energy of light to make ATP. So that's the 3 ways in which ATP gets made inside of cells. In our cells, well over 95% of our ATP is made by oxidative phosphorylation. Very little gets made by this mechanism. Yes? The second one was called oxidative phosphorylation. Yes?
I just wanted, so substrate level phosphorylation happens when a high energy molecule transfers what?
Transfers a phosphate to ADP to make ATP. That's substrate-level phosphorylation. And we will see reactions. In fact, there's another one later in glycolysis that's also a substrate-level phosphorylation. But that's not where the majority of ATP is made in the cell. The majority of ATP that's made in the cell is made by oxidative phosphorylation that we will see. All right. Well, at this point, we have 2 molecules of 3-phosphoglycerate. The enzyme that catalyzed this was called phosphoglycerate kinase. There's phosphoglycerate kinase. All right. There's 3-phosphoglycerate. I told you that there's a molecule like we've talked about before, but I haven't brought it up yet. That comes up in the next reaction. Now, I need to set this up for you. This is kind of cool. Okay. The next reaction is catalyzed by this enzyme called phosphoglyceromutase. All right? I want to tell you a little bit about what mutases do. Okay? So whenever we hear the name MUTase in an enzyme, it describes a mechanism that the enzyme is actually using. Okay? Well, what's the mechanism? Here's what happens. We look at this and we say, well, this enzyme is simply taking this phosphate and it's moving it from right here on position number 3 over here to position number 2. And that's not what it does. It just looks like that. Okay? Well, how does it get from here over to here? The enzyme uses a little sidestep. It makes an intermediate, again, up here, that I'm going to describe to you. The intermediate that it makes is as follows. It starts with 3-phosphoglycerate, and it puts a phosphate on position number 2. Now, that makes a molecule that you have heard of before. It's known as 2,3-bisphosphoglycerate, or 2,3-BPG. Then, it's got phosphates on positions 2 and 3. Guess what it does? It takes the phosphate off of position 3, and now it's left with position number 2. So 2,3-BPG is an intermediate in this reaction. It's one of the few stable intermediates that's produced in enzymatic reactions. That turns out to be really, really useful for us because this enzyme doesn't hold on to 100% of these and convert 100% of these into 100% of these. A small percentage of the 2,3-BPGs escape. Well, that turns out to be pretty cool because what does 2,3-BPG do? You may recall it binds to hemoglobin, and hemoglobin switches into the T state and lets go of oxygen, right? Well, and I told you at the time that cells use 2,3-BPG as a flag that says metabolism is rapidly going on here. Well, how do we get any amount of 2,3-BPG if only a small amount of it is released at any given time? We have a lot of metabolism going on. So a cell that's burning a lot of energy will be making a lot of this and making some of this and releasing a little bit of 2,3-BPG. The more this pathway is running, the more 2,3-BPG is released, and the more hemoglobin knows where to dump its oxygen. It's a perfect flag. When we have very little metabolism going on, there's essentially no 2,3-BPG that's released because, remember, it's only being released at a very low efficiency. And so hemoglobin doesn't drop off any oxygen there because there's not much metabolism that's going on. Okay, so this intermediate turns out to be really cool, and it really is serving as that flag that tells the cell metabolism is occurring rapidly here. Questions about that? Okay. Well, the next reaction is a pretty straightforward reaction. We actually pull water out of this guy. Okay? So 2-phosphoglycerate, which is called 2PG, by the way. Okay? 2PG, all right, loses water and makes this double bonded molecule here. Right? This guy, phosphoenol pyruvate, also abbreviated PEP, okay, you can call it PEP, is one of the highest energy intermediates inside of cells. It's very, very energetic. It's full of energy. Now we're going to see in the next reaction that the energy of this molecule is going to be used to make ATP. But there's something else that I'll show you that happens with this also. Okay? So we see what's happening with this guy here. The enzyme that catalyzes this is called enolase. And we have PEP. Now, we're coming up for the reaction in glycolysis that I like to call the Big Bang. Okay? When we think of the Big Bang was the explosion that started the universe, this Big Bang is called a Big Bang for another reason, and that's because it's extraordinarily energetically favorable. It's extraordinarily energetically favorable. Okay? What does that mean? Okay? First of all, let's look at the reaction. Pyruvate kinase is the enzyme. we have a high-energy phosphate, and that high-energy phosphate is transferred to ADP to make ATP. So this is another substrate-level phosphorylation. Okay. Now, here's the cool feature about this reaction. All right. The cool thing here is that there's enough energy in this guy to literally make 2 ATPs. There's only one phosphate, though. Right? So here's this guy. It's just loaded with energy, but it's only got one thing it can donate. What happens to all that extra energy? It's released as heat. One byproduct of metabolism is heat. Well, this reaction right here generates about as much heat as any reaction that occurs in your body. I want you to think about this the next time you go out exercising. The next time you go out exercising, what are your muscle cells doing? Well, they're generating ATP. They're generating ATP by burning down sugar. And when they're burning down sugar, they're running this reaction a lot. And when they run this reaction a lot, what's this reaction going to produce as a byproduct? Heat. And what are you going to do? You're going to sweat because you get hot. The reason you get hot when you exercise is you are making all of these processes, which are energetically favorable, go forwards. But this one in particular is very energetically favorable, and you're generating a lot of excess heat. Clear as mud? Very good question. So the delta G for this reaction, if we started out with equal concentrations of products and reactants, the delta G would be very negative. That means that the delta G0 prime is very negative, very energetically favorable in the forward direction. And there literally is enough energy in this reaction to make a second ATP. You guys don't look like you're very energetically reactive at all right now. You look like you're just, God, it is Friday afternoon. Wild plans for the weekend? Studying for bio. Oh, I like to hear this. This is good. So, A grade right there. Okay. Well, let's finish a couple things here, and then we'll have a song. How about that? And we might even finish a little bit early today. How's that? Was that a no I heard? Okay. Well, one of the things I haven't talked about was the very first thing I skipped over. So now we're at a point where we can start to understand that very first thing. Okay? Pyruvate. We've just made pyruvate. Pyruvate turns out to be a really, really important molecule in cells because pyruvate is a branch point between several things. When I say a branch point, I'm talking about a molecule that can go in multiple directions. Okay? Well, one of the directions that pyruvate can go is that pyruvate can be very, very easily converted into the amino acid alanine. All we have to do is swap an oxygen for a nitrogen, and we've got alanine. Very readily interconverted. There's some other things that can happen to pyruvate, and that's what's shown in this slide right here. Okay? Here we are. Here's pyruvate. All of glycolysis has been scrunched up here so I can show you what's happening down below. All right? Pyruvate has 3 different fates, okay, with respect to oxidation, and it depends on the cell in which we're talking about. Let's think about this, okay? So let's start with us, okay? As we're sitting here and we're breathing, and we've got plenty of oxygen, and our oxygen supply is high, our pyruvate is being taken, and it is being converted into acetyl-CoA, and it is oxidized in the citric acid cycle. That's where oxidative phosphorylation ultimately comes from, all right? So one step is to convert pyruvate into acetyl-CoA. That occurs in any cell that's aerobic and has oxygen. That could be our cells. That could be a yeast cell. That could be a bacterial cell. As long as it's an aerobic cell, when oxygen's present, that's what's going to happen. Okay? Well, what about cells that don't have oxygen? Well, we have that happen in our body as well. When we're exercising, our muscle cells, especially if we're exercising heavily, can use oxygen faster than the blood supply can deliver it. We go anaerobic. Okay? When that happens, pyruvate is converted into lactate. Notice lactate is not the same as lactose. Lactose was a sugar. Lactate is a 3-carbon molecule. Why does this happen? Why does this happen? Well, that's partly illustrated on this screen right here. All right? It happens because cells need to regenerate this guy right here. Why? First of all, they have a limited supply of it. If everything they have is in the form of NADH, nothing will be in the form of NAD. Well, why is that of concern? Look what we did up here. During that oxidative reaction with G3PDH, we converted NAD into NADH. In doing so, we no longer have NAD. If we don't regenerate NAD, what's going to happen to glycolysis? It's going to stop right at that step. And if we need energy, that's not good. The muscle cell is working so hard, that's why it's run out of oxygen. So we want to keep that muscle cell going. And the way we keep that muscle cell going is by doing fermentation. Your cells are doing fermentation anytime they are regenerating NAD like this. Okay? The word fermentation we frequently associate with alcohol. Okay? We're not making ethanol here. We're not making beer. We're not making wine. Our Friday evenings would be a lot different if we did. Hey guys, let's go out running, right? Wouldn't that be great? Whoa, whoa. And the further you run, the worse you got, you know? But you'd be so fit. I mean, wouldn't this be great? Okay. So you're fermenting when this happens. I never thought about that before, but it's kind of cool, right? Okay? You're fermenting when this happens. So now these NADs can go back and be used to keep glycolysis going. This process is really inefficient. But it's necessary. Without this, you stop glycolysis and you just killed that muscle cell. So it's very important, but as we will see later, it's a very inefficient process. Okay? Well, so that's what happens in our cells. This will happen in any animal. Any animal will have this process going on. And lactic acid, when it accumulates, some people think that it's what causes muscles to be sore after heavy exercise. That's disputed. I won't say one way or the other. Okay? But the point is that when you exercise heavily, you do make a fair amount of lactic acid. Yes, Lisa?
When they talk about increasing your lactate threshold, is that because your body produces more NAD?
So your lactate threshold really is how much you can produce and handle. And that'll actually get into something called the Cori cycle that I'll talk about in a bit. So let me save that for then, okay? Okay. Well, what if I'm not an animal cell? What if I'm a bacterium or a yeast? Okay? This is where it gets good. And it gets good because they don't make lactate, which is, all, by the way, is the same as lactic acid. All right? Instead, they convert pyruvate in a couple of steps to ethanol. And they're doing it for the same reason. They're trying to make NAD. And when you make beer and you make alcohol, I know there's some fermentation people in here, you have to make sure that you close the bottle so that they don't have the oxygen, because if they have the oxygen, they're not going to make ethanol. They're going to make you a real mess is what they're going to make you. So you close the bottle, you make sure they don't have oxygen to cause this to happen, and when this happens, they make ethanol. They're doing it to keep glycolysis going so they can stay alive. Okay. Makes sense? Everybody understands the different things. So acetyl-CoA, when oxygen is present for any aerobic cell, okay? Lactate over here, animal cells when they're out of oxygen, ethanol when we make, and when bacteria and yeast are doing it. Okay. That's pretty important, pretty cool. I'm going to talk a little bit about regulation here, but I'm going to talk more about regulation when I talk about the synthesis of glucose, because it turns out that the synthesis of glucose is in parallel, or I should say anti-parallel, with that of glycolysis. Here's the things that you've already seen that's here. Okay? I want to point out a couple things to you. One is right here. Okay? Fructose 6-phosphate going to fructose 1,6-bisphosphate. This was the enzyme PFK that I told you was very important. I said it was probably the most important regulatory enzyme in glycolysis. And I said regulation is really critical because if we don't control this pathway, we're in trouble. Well, when do we want to stop this pathway? The analogy I gave in class was if I have a fireplace in the middle of summer, I don't want to be burning my fireplace and wasting wood, right? I'm just putting out heat that's no good. I similarly don't want to be oxidizing my glucose if my cells are full of ATP. I wouldn't want to waste my energy like that. So look what happens. When ATP accumulates, what does it do? It allosterically inhibits PFK. It allosterically inhibits. So here, PFK is reading, as it were, the energy level of the cell. It's reading it. And now it knows, okay, this guy isn't exercising, he's not getting out and doing his thing. I better slow down glycolysis or we're going to waste a lot of energy. Okay. This guy is inhibited by ATP. Okay. Now I'm going to say more about both. This is pyruvate kinase, by the way. I'm going to say more about pyruvate kinase and PFK later. The very first enzyme is called hexokinase. There are 3 enzymes in glycolysis that are regulated. And it's a very unusual pathway in this respect. Most pathways have 1 regulation step. This guy has 3. 1 in the first, 1 in the third, and 1 in the last enzyme. Okay? Now there's reasons for why that occurs. They won't be apparent if I talk about them today. But I just want to plant these in your head that these 3 are the regulated steps. And I think you'll see when I talk about the synthesis of glucose why these make some sense. The third one is, okay, so hexokinase here, PFK here, and pyruvate kinase down here. Okay? Now, it turns out that both PFK and pyruvate kinase are inhibited by multiple things. We're going to see some interesting interplay with that in just a bit. Okay. There's pyruvate going to lactate. I'm not going to make you memorize that enzyme name. Don't worry about that. Okay? But you can see the reaction that's going on. Here's a ketone. The ketone is getting reduced to an alcohol. It's taking electrons from NADH and making NAD. Here's alcoholic fermentation. Okay? It's shown down here. Now this one is, I find, especially appropriate for a Friday afternoon. Okay? The reason? Well, here's pyruvate. Here's how the bacterial and yeast cells make ethanol. Okay? But there's another thing in here. The other thing in here is this guy called acetaldehyde. Okay? Ethanol is not made directly from pyruvate. It's made from acetaldehyde. And this enzyme, yeah, I think would be a good one to know, alcohol dehydrogenase, is an enzyme that you have in your body. You have this enzyme in your body. Why can't you go through this process? Well, you can't go through this process because you can't do this step. You can't do that. If you could do that, we would have that running happy hour, right? Okay. Well, then the question is, well, why in the hell do you have this enzyme? The reason you have this enzyme is because your body sees this guy as a poison. And so what it's doing is it's making the reverse reaction. It's making acetaldehyde. And when it makes acetaldehyde, you get yourself a hangover. Okay? Now, bacteria and yeast, of course, don't have a hangover. They're making it go this way. I suppose if you let their ethanol accumulate, they probably would have the equivalent of a hangover. Right? Because that'll mean that this guy will also accumulate. But that's what's happening here. And again, this figure is simply showing you that what they're doing is they're making NAD, and that NAD is being used to keep this reaction going. This is the one in glycolysis that needs the NAD. Okay, questions about that? No? Okay. What do you say we have a song and we finish the week? How about that? All right. You got to sing loud, though, otherwise I'll go back and lecture. All right? Loud singing is required. All right. It's called The Sound of Glucose. It's to the tune of a few of my favorite things. Aldehyde sugars are always aldoses, and if there's a ketone, we call them ketoses. Some will form structures in circular rings. Saccharides do some incredible things. Onto a glucose, we add a P to it. JTP energy ought to renew it. Quick rearranging creates F6P without requiring input energy. At a high rate, add a phosphate. FPFK. F16BP is made up this way so we can run and play. Da 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 da. We're not done yet. Okay. Aldolase breaks it and then it releases DHAP and a few G3 pieces. These both turn into 1,3-BPG, adding electrons onto NAD. Phosphate plus ADP makes ATP, while giving cells what they need, energy, making triphosphates a situation of substrate level phosphorylation. 3-BPG, 2-BPG, lose the water. PEP gets a high energy state just to make pyruvate. Da, 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 da. So all the glucose gets broken and bent. If there's no oxygen, cells must ferment. Pyruvate, lactate, our cells hit the wall. Some lucky yeast get to make ethanol. This is the end of your glucose's song, unless you goof up and get it all wrong. Break it, don't make it to yield ATP. You'll save your cells from futility. Da, 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 da. All right, guys, have a fun weekend.